Today, I'm super excited to have Brenda Oppert here in our Zoom virtual studio. She's in Kansas right now, but I'm in Vancouver, Canada, and I hope everyone out there is safe and healthy. So like I said, we're having 5 of these talks over the next 2 months, and Brenda's is the first one. So this is sort of, you guys are kind of a bit of a guinea pig here in terms of doing this webinar presentation, but I hope it goes smoothly. And I think Brenda's got some super exciting science to share with us on her insect genomes. But before I introduce Brenda, I just want to give a very brief 6 or 7 minute introduction to Dovetail, because there could be people on the call here that are either new to Dovetail or new to de novo assembly, and I just want to make sure we're all aligned so that you can appreciate Brenda's presentation even more. So Dovetail is a proximity ligation company. So our sort of core technology is all to do with proximity ligation. And what is proximity ligation? Basically, it's chopping up the chromosomes of an organism cell in situ and then ligating them back together is really, in simplest terms, what we're doing. And you can see here, this is obviously a cartoon of a chromosome, and you can see that when we chop up a chromosome and religate it back together, there's going to be points on the 3D chromosome that are closer together and more likely to ligate than others. So the circle I've drawn there shows you points on the chromosome in 3D space that are very close together, and those have a much higher probability of ligating than the line that I've drawn at the top, which shows, here we go, my pointer, but this shows 2 points in 3D space that are much farther away from each other and are less likely to ligate. So you get this curve of proximity ligation events. The ones at the top of the curve are far greater in abundance and closer together. And as you go down the curve, they get farther and farther apart until you get to the telomeres. And so this is a distribution. This is a typical HiRise distribution plot. HiRise is one of the proximity ligation protocols that we use. And this is a great way of not only profiling the biological content of the chromosome for epigenetics, but also it's a great way to create pseudo-long reads that can be used to scaffold a genome assembly. Just very briefly going through the timeline of Dovetail. So we started in 2013. This company arose out of Ed Green's lab at UC Santa Cruz, and he developed Chicago, which is an in vitro proximity ligation technology, unlike HiRise, which is in situ. And Nick Putnam wrote the software HiRise, which we still use today for our scaffolding software. And they were very successfully scaffolded the American alligator assembly. But however, they were very successful, but they did not get it to chromosome scale because Chicago is not an in situ HiRise, it's done in vitro. So to get up to chromosome scale, we had to launch our Dovetail HiRise methodology, which we did in 2015, and that is an in situ version of proximity ligation. So we are, after 2015, we were able to start assembling full chromosomes. Last year, we launched our newest proximity ligation technology called OmniC, which is, unlike HiRise, OmniC uses a random endonuclease. HiRise uses a restriction enzyme. And I'll explain the benefits of that in a few minutes. We're still using HiRise, and we worked on the narwhal. That was one of our first projects that we did. And then this year, we're now launching phased assemblies. So we've successfully phased human assemblies using OmniC. And finally, 2020 and beyond, we're hoping to now move this into true diploid assembly. We successfully assembled the tuna. Both maternal and paternal chromosomes were separated and assembled independently. This is the pipeline that we use today. And basically we're using PacBio, deep PacBio sequencing with WTD-BG2 as the assembler. We scaffold with OmniC and HiRise. We do a full annotation of the genome, and I think Brenda just got her genome annotated like 2 days ago. So hopefully she has some updates on that for her assemblies. But we can do the entire annotation process at Dovetail and give you a publishable assembly. By the way, there's a Q&A box. I should have mentioned this. There's a little Q&A button at the bottom of your screen. So feel free to just click on that and type in any questions you have, because everyone is muted right now. And then we'll get to the questions by the end of the talk. So feel free to type in as many questions that you'd like. So there's several major challenges with de novo assembly, as you probably can appreciate, shown on the left. But these challenges, we can overcome them all pretty much. I'd say in the 1,500 assemblies we've done to date, there's probably been less than 5 that we essentially couldn't really handle because of the challenges, the inherent challenges of the assembly. So it's extremely unlikely that we can't assemble a genome. In fact, I think it's less than 5, probably like 2. But on the right is our tree of life. And I think the tree of life is a graphic that's available on our website, and you're free to browse that. These are genomes that our customers have said it's okay to publicly talk about. So there's only 400 on there out of the 1,500, but it gives you a really good representation of our experience, that we've built genomes from the most challenging taxa you can imagine, from especially marine invertebrates, insects, plants. So we can overcome the challenges shown on the left because we've done so many projects from virtually all taxa. Very, very briefly, how does OmniC work? And this applies actually to proximity ligation in general. You basically cross-link the proteins in the nucleus. You fragment the chromosomes, and in OmniC's case, we're using an endonuclease. You're chopping up the chromosomes, then you're ligating them all back together. The stuff that's the points that are closer together are going to ligate more frequently than the ones that are farther apart. So you get that nice distribution that I showed you. We reverse the crosslinks, freeing up the DNA, and then sequence those molecules on Illumina sequencer. And then that data goes into our HiRise pipeline. And HiRise is essentially taking the scaffolding sequencing data plus your draft assembly that you built with PacBio, and it essentially tries to build super scaffolds up to chromosome scale. And you can see the data on the left here is what your assembly looks like before going into HiRise. So going across that diagonal, each of those little squares is a scaffold, and you can see that there's thousands of them. After HiRise, out pops your chromosome scale assembly. Each of these squares is in fact a chromosome. My last slide is just showing you the benefits of OmniC. So why is OmniC sort of the latest and greatest thing? It's because OmniC uses an endonuclease, unlike traditional Hi-C that uses a restriction enzyme. And what we see with OmniC data is that the contiguity isn't really that much different than if you use a restriction enzyme. I think it is a bit better because it makes sense that you're covering more of the genome. You don't have any restriction enzyme deserts. But what is really powerful with OmniC data is because we're getting very even coverage across the entire genome using a random cutter, we can capture almost all of the SNPs in the genome. And by doing that, we can then phase the genome. And I think that's the crucial difference. You know, it's just more even coverage, which enables us to phase, which is again, one of the polling questions that is up on your screen. So with that, I think I've done my time. I wanted to get it over to Brenda as fast as possible to learn about what Brenda is doing with her insect genomes. And Brenda's based in Kansas State, and she's a research molecular biologist with the USDA. And Brenda's done several projects with us over the years. It's been really fun working with Brenda, and I'm really excited to hear her latest findings. So with that, Brenda, I will turn it over to you.
Okay.
And yeah, see your screen fine.
See if I can get the slideshow going. Okay. Does that look okay?
That's perfect, Brenda.
All right. So I wanted to thank Mark and also Dovetail for the opportunity to present to you today in the first of the webinar series. I work for the Agricultural Research Service. It's a lab in Manhattan, Kansas. And this is an aerial view of the grain bins on our campus. I work in the Stored Product Insects and Engineering Research Unit, where we partner with farmers and food producers to limit the damage to stored grain and grain products. We have stakeholders that include operators of flour mills and warehouse managers. The reason that we're interested in working in this area is that insects can destroy up to 30% of food in developing countries, and the economic impact is large. Effective control is threatened by the deregulation of fumigants, and insecticide resistance is the most pressing problem, and we really need sequenced genomes in order to work on these problems. So this is a little video of the red flower beetle. These are some of the worst of the stored product pests that we study. These tiny little beetles are often found in flour products, and you may have actually seen them in your pantry if you're unlucky. They are problematic worldwide, but they also have served as a model for coleopteran genomics and have been very useful to a number of groups. So I'm just going to start talking about this insect. It's the latest one that we're working on. But just to give you some background that I'll talk about in the rest of the talk. Tribolium castaneum has a complete genome sequenced. It was actually the first stored product insect and the first coleopteran and the first agriculturally important insect to have a genome sequence. It was a mammoth undertaking at the time. It was entirely with Sanger sequencing, very expensive. And we used a highly inbred strain, and that was published in 2008. So the most current version of that assembly is TCAS 5.2. And while this genome has been highly utilized for genome studies in a number of organisms, it lacks about a third of the genome sequence. My colleague, Marseille Lorenzen at North Carolina State University, has been working on this insect probably all of her professional life and refers to them as her babies. We partnered to see if long read and long range data could improve this genome assembly. So we started with sequencing on the PacBio Sequel 1, and we got 150x coverage, and we were pretty happy with that with 4 SMART cells. But when we tried 1 SMART cell on the Sequel 2, we actually got 350x coverage. So that gives you an idea of how much improved the Sequel 2 is in sequencing right now. We scaffolded this with the Dovetail HiC and HiRise. And we were able to, so the new assembly is shown on the right, and we were able to get a complete assembly. This is a 200 million base para genome, and the scaffold N50 was increased, and as expected, the contigs and number of contigs and number of scaffolds in the scaffold L50 was decreased. So that just gives you an idea about how organisms that have been previously sequenced can benefit from this approach. So we developed a reference quality insect genome roadmap, and in working with Dovetail, our first challenge was to get high quality long genomic DNA that was needed for the long read sequencing. And we were able to achieve that finally, and then working with Sergey Koren, we developed KNU as our assembly algorithm, and then obtained long-range data when partnering with Dovetail. And we did that through Chicago and HiRise library, and using the HiRise software. We most recently, as Mark said, we've partnered with Dovetail on annotation of these genomes, and I will talk a little bit about that, as well as some of the assessments that we do just to look for quality in the genome assembly, such as BOSCO and omics box. So our first insect that we assembled using this roadmap was Rhyzopertha dominica. It's a lesser grain borer, and it is a major pest of stored grains. The immature stages of this insect develop within the grain, and that makes it difficult to know that you've got an infestation until it's too late. On the left, you can see that the X-ray shows these larvae feeding inside the grain kernels. The problem, as I told you, with these insects is they develop resistance to insecticides. Because they are it's just the nature of the way that they are, of the environment. They're reared in close, I mean, they develop in close environments. And so resistance is always a problem. This insect is notorious for developing resistance to the control products that have been effective in the past. And one of the reasons that we need a reference genome in order to evaluate these resistant populations. And a number of control products have been effective, but one by one, we're losing these control products to resistant insects. Sorry. So the genome sequence was obtained from the pupa stage, and this is a pupa that was used for extraction of the genomic DNA. We obsessively inbred this insect for more than 2 years prior to sequencing. And I say obsessively because we don't think that this is really necessary. We have partnered with Spencer Johnston at Texas A&M to provide accurate measurements of the size of the genome. And this genome was actually pretty similar to what had been published in the literature, the size. We sequenced genomic DNA on a number of platforms, but the draft assembly was with PacBio RSII data by Tim Smith, and we made multiple assemblies until we had a good draft assembly with KNU. So the draft assembly had 1,861 scaffolds and a BOSCO score of 99%. But Dovetail was able to reduce the number of scaffolds and increase the N50 with Chicago HiRise scaffolding. We did take this assembly and used Illumina data to obtain what we call a hybrid assembly in an attempt to increase the accuracy. But the accuracy of PacBio data now is better, and so we no longer do this. The HiC scaffolding provided a final assembly of 140 scaffolds with an N50 of 53.6 megabases. And this is the linked histogram for the HiC scaffolds, indicating that essentially we have scaffolded chromosomes for this insect. Skip the slide. This is the annotation, the first annotation that we've completed with Dovetail, and I just got this data last weekend and haven't received the data files yet, but I thought I would give you some information on the pipeline that Schwen Ho uses for genome annotation. After masking the genome, gene prediction is a rigorous process of training prediction with Maker, SNAP, and Augustus. And then AED scores are provided for quality assessment of the predictions. And these are the AED scores that we received for this annotation. It indicates that most of the predictions are high quality. This is the excerpt from the report on the left. Gives you an idea of kind of information that you get back. We had a total of 24,720 genes annotated in this genome. And we had already done some preliminary assessment of the genome and had done masking. So our masking results are on the right, and you can see that they are pretty much in agreement with the results that we got back from the dovetail annotation. So the next insect was the rice weevil. It's also an internal feeder, very problematic in the immature stages. Weevils are problematic not only in stored products, but also in the field. And in fact, one of the most economically damaging weevils is the boll weevil in cotton, which producers have spent a lot of money to eradicate. So the genome sequencing, and this time we inbred for 10 generations. The genome size was similar to what was found in the literature. And we sequenced on the RS2 with 44x coverage. So the canoe assembly of the PacBio data indicated that the assembly had a BUSCO score of 98%. The Chicago HiRise and HiRise scaffolding provided a final assembly of 498 scaffolds with an N50 of 45.6 megabases. And this is the link density histogram that indicated, again, we had mostly chromosome level scaffolds in the final assembly. So the next insect that we worked on was the yellow mealworm. And this is not really a problematic stored product pest. It's more of a model that we use for biochemical studies for many years. It can be problematic in poultry houses. But I began this project as a proof of concept to see if we could get a draft assembly and scaffold from the same insect to avoid inbreeding. And this insect has a really long developmental time, so it's just not possible to inbreed. During the draft assembly, I joined forces with Aaron Dossi at All Things Bugs to provide genetic resources for insects as food, a project that was funded by DARPA, funding to All Things Bugs. So this was a new area for me to work in, because up till now I've just been trying to kill insects, and now I'm going to try to make them better. So working to improve insects as a food crop has been provoked by the food insecurity that has been driven by climate change. And this report by FAO and other world organizations warns that in just 10 years, we may have difficulty in addressing hunger unless we change the way we approach traditional agriculture. And the problem is the lack of sustainable protein. The world population is predicted to increase to 9 billion by 2050. And we already devote 70% of agriculture land and 30% of the total land in the world to raising livestock. The demand for protein is increasing as the population increases, but we will not be able to increase the amount of protein through traditional livestock to meet the demand. So this is a summary of data that will hopefully convince you, or at least make you think about it, how much more efficient insects are at converting food to protein. In the summary of data of publications, we find that insects are an excellent alternative source because they require lower inputs of feed and water, and the amount of land footprint required to rear these insects is much smaller. in addition, another benefit is that insects have a lower impact on the environment. In this study from PLOS One, mealworms were determined to have lower impacts in the environment with lower emissions of CO2. And since insects can be reared vertically in contained environments, the amount of environmental impact can be reduced even further. And you may not have thought about this, but 3/4 of our food comes from just 12 plant and 5 animal species. The lack of diversity in agriculture could greatly benefit by incorporating insects as food crops. So back to the Tmo genome. We estimated the genome size had been estimated in the literature at 509 megabases, but the actual size is much larger. It's more towards 800 megabases. So to get, so like traditional livestock, we need genome sequences so they can be improved for food applications. And so we developed a new extraction protocol based on a kit from Omega Biotech. And the genomic DNA was sequenced on the RS2, but we had problems with the assembly. And Sergey was able to finally take over on this and get us a draft assembly, but he said that the compute time was similar to what he had used for a human genome. And so, and this is the information on that extraction protocol. I'm happy to send that to you if you'd like to have that. The problem is with the assembly we found in the literature was this report by Davis and Wyatt that described a satellite in this insect. The satellite was 142 nucleotides and it's highly homogeneous. When I blasted the nucleotide sequence of the satellite to the PacBio reads, I found the satellites peppered in the reads represented by the pink segments that you see. This satellite comprises 60% of the genome and is found in every chromosome. And this is why we were having a problem with this assembly. But still, we were able to get, using long read and long range data, we were still able to get an assembly and we're able to get scaffolded data. But still, we only have about 60% of the genome assembled, although the BUSCO scores are in the 98% range. And so it indicates we have the coding sequences, we just don't have a lot of these repeat structures that we are going to continue to work on. And this is the LinkedIn histogram showing the chromosome scaffolds. But we are going to attempt to resequence this insect with a PacBio high fidelity reads and incorporate the Dovetail Omni-C to see if we can pull in some of these other additional regions that may be important in regulatory regulatory regions that we may be interested in for resistance and other information. So I'll just end with the results that we got on the cricket assemblies. We have sequenced both Acadia domesticus, which is the house cricket, and I never can say this insect, Grylodes sigillatus, the banded cricket. These are two crickets that are commonly used for food products. They have been used mostly as feeder insects for reptiles and birds in the US, but around the world they are a common food product. The assemblies that we got, even though these are very large genomes, much larger than the ones we've been dealing with previously, the assemblies that we got are very good. We used Sequel 1 for the draft assemblies and Chicago and HiC data to scaffold. The N50s for both insects were more than 200 megabases. And these are the HiC histograms that suggest that we have scaffolded to chromosomal level and both insects. So I took the 11 largest scaffolds in the crickets, and I compared those to the 11 largest scaffolds in the grain borer, and looked at the BUSCO scores in sequences from those 11 scaffolds. And these data give me confidence that we have mostly chromosome level scaffolds in these largest scaffolds. Now, the reason that we sequenced both the house cricket and the banded cricket is that the house cricket ADO is susceptible to a densovirus, and in fact was decimated. The colonies of the house cricket were decimated by this densovirus, and so they brought the banded cricket in because it was resistant to this virus, and to use it as a replacement in cricket farms. We used KRAKEN to evaluate the scaffolds for the metagenomic sequences, and we know that from Sergey's talk at Plant and Animal Genomics that when you do the assemblies with PacBio, you also can assemble the metagenomic sequences, and in fact that's what happened with these crickets. The banded cricket, when we looked at the metagenomic sequences, they were about half of the scaffolds contributed to by the metagenomic sequences, and mostly those were from bacteria. But about 3/4 of the scaffolds from the house cricket, ADO, were metagenomic sequences, and there were more virus sequences than bacteria. So this suggests that there could be a genetic component to the low number of viruses that we see in the banded cricket assembly, and could explain why the banded cricket is not susceptible to densovirus. So we are going to be looking at that more closely to see if we can determine what that is. We also noticed that the cricket genes are much larger than the orthologs in the beetle Tribolin castanium. I just pulled out the BUSCO genes, the length of the BUSCO genes from the crickets, and compared them to the beetle. And in both cases, we see, this was sorted for ADO just for ease of viewing, but in both cases we see that the genes are larger than those, longer than those in the beetle Tribolium, but in some cases they're a lot larger, very much larger. And so we looked at this a little bit to try to understand why they are so much larger. And in fact, it seems that it's because of the intron size. This paper describes that in orthopterans, the introns are larger, and they found that there is a positive correlation between the amount of transposable elements in the genome and the genome assembly size. But we know that we have a lot of TE content in these beetles, and so we think there could be other explanations, but we're going to be looking at this a little bit closer before we release this genome, these genomes. This gives you an idea about what these repeat structures are in the cricket assemblies. And in both insects, they were similar as far as the categories that you see. And about half of these are in the unknown or uncategorized category. So the most prevalent repeats that we find in both crickets are of the category line RTE both B. And these are, let's just go to the next one. These are the most abundant TE families found in mammals, and it was proposed that they were introduced as foreign DNA via ancient horizontal transfer. It was also proposed that in insects, horizontal transfer of these elements is a major driving factor for insect molecular evolution. And our data in crickets support this hypothesis. So I want to end by going back to the reference quality insect genome roadmap to reiterate that each one of these steps builds on the other. So it's really important that you start with really good, high quality, long genomic DNA. That's the most important step in order to obtain that from your insect. And then it's also important to obtain long read data. We've used PacBio, but we are going to start working with Oxford Nanopore to see if we can incorporate that some into our assemblies as well. The draft assembly of the long read data, we prefer to use Canoe because it's less compute intensive, but it gives us good draft assemblies. We think it's essential that you obtain long-range data, and we have been very pleased with the results that we've gotten from using both Chicago and HiRise in our scaffolding. The annotation can sometimes slow down progress to release these genomes, and it's been one of our problems, and so we were very happy when Dovetail offered this service, so we can hopefully speed up the release of these genomes. And then there's a lot of tools that you can use for doing the quality assessment of the genome. So I'll end by thanking a lot of people that were involved. These are the major players in the information that I gave you today.
I'll just read you a few questions here, Brenda, and
okay.
Because they're specific to your projects. So the first one is from Nima is, what was the sex of the individual sequenced for the first assembly?
They're all males, and all of the assemblies I talked about today, they're all males.
Okay, thank you. The second question is, okay, so is the new T. castaneum assembly based on inbred male or outbred?
Inbred. It's the same strain that was used for the original sequencing project called Georgia 2 or GA2, and that strain has been maintained in the lab for the genetic studies. So we just selected, I think it was a group of like 10 individuals from that. It was a pool of insects.
Okay. Did you, also from Nima, did you run Busco on predicted genes after annotation?
Yes, actually that was part of the report, that I didn't give you that information, but yes, the Busco is also given to us as part of the report.
Okay, great. What about, that was Nima again. So, I'll go back to Nima if we have time, Nima. So Fidel asked for, let me try and get this right, hemimetabolan insects, which do not have pupae. Do you know if some differences are observed among instars in terms of DNA quality after extraction?
I don't know specifically, but I can tell you that every insect is different. Every insect has a extraction protocol that works well for it. When we did the first two assemblies with the lesser grain borer and the rice weevil, we were extracting DNA using a Zymo research kit, and that worked well. But when I got to the Tenebrium molitor project, that didn't work well for us, so we had to switch and try something different. We tried a number of kits, and we liked the Omega Biotech kit, especially for larger insect samples. It has worked well for us as well. But I'm working on some lepidopterans right now, and it's a different ball game with them. Having to try some different things to try to get DNA that's not damaged, and DNA that's long for those studies. So every insect is likely going to be different. You can start with some published procedures, but then you may have to tailor it or try something different.
Okay, thanks. Thanks, Brenda. Do you see any correlation between GC content and gene length?
I have not looked at that, but in the paper that I cited, I don't think that they found a correlation between that. It was mostly between the TE content, and certainly I think that's a factor. But is that the only factor? I don't know.
Okay. Some of these, I want to right now just address Brenda's question since she's with us live here. Okay, so here's one. Can you talk a bit more about the challenges of working with large genomes in general? I work with insects that have 8 gigabase genome size. I guess that's kind of a question for me then, because Brenda, yours aren't that large, right?
No, but we've considered working with some of the larger ones, and I think you know, the experience with the cricket was so really easier in some respects than certainly those tenebrionid beetles, the red flower beetle and yellow mealworm. Those are both still problems because of the repeat structure. But with the crickets, if you get sufficient long read data and the long read data is good, and then you use the scaffolding, I just don't foresee that it would be any more of a problem.
Okay. Yeah.
Yeah, go ahead.
Yeah. We're going to have a lecture. I believe it's it's tomorrow on super large genomes. So you might want to attend that webinar. We have at Dovetail done several very large genomes, including the sequoia and the redwood. Obviously, they're not insects, but you know, the principles are the same. It's one of the challenges of de novo assembly, and we've been very successful in assembling those genomes, very large genome sizes. So I encourage you to come to the talk tomorrow, where Stephen Salzberg is going to be talking about very big genomes and the tricks and techniques to use for those. Let's see here. Okay, here's one for you, Brenda. How do you remove bacteria or virus sequences from the assembly?
So that's a good question. That's what we're working on right now. We are going to hopefully be able to use the information that we get from KRAKEN in order to identify those scaffolds and be able to just separate those out of the crickets. And actually, I was talking with Schwen about this. We were supposed to have a conversation this afternoon about doing that before he moves forward on the annotation project. So we get the identification of those scaffolds from KRAKEN, so we should be able to write a program in Perl and be able to do that.
Excellent, excellent. And just for everyone on the call, Schwen is our annotation expert. So he is a dedicated bioinformatician that does all the genome annotation. So here's another question for you, Brenda. Are there any novel innate immunity genes that protect the insects from viruses? That's an
well, certainly that's what we're going to be looking at. Immunity genes will be, you know, one of the categories that we'll be looking for. We have a lot of transcriptome data from the house cricket. We actually sequenced all life stages, and that paper came out recently. And so we have a great deal of data on transcriptome in that insect, but unfortunately we have nothing in the banded cricket. And so I'm getting ready to do some transcriptome sequencing in that insect as well. And that may give us some clues as to what sort of genes could be involved on viral challenge if we do some kind of a comparative sequencing study. It's difficult to work with the viruses in the lab Aaron tells me, because they are so, they have to really keep them separate away from the house crickets, because they really can wipe out a colony. So there's some logistics involved in that, but certainly, yeah, I would think that immunity genes would be one of the targets to look at.
Mm-hmm. Okay. Here's a question specific to insects, which a lot of people probably wonder. Do you prefer pupae due to easier extraction or something else? Adults might have more DNA. So can you talk about the different developmental stages and what you have been most successful with using?
Sure. So we prefer to use pupae in the stored product insects because the extraction procedure is easier. The tissue is softer. We don't have the hard cuticle that gunks up the filters and things. We get probably optimal amounts of genomic DNA from the pupae. So that's, in most cases, the genomes that I presented today were with pupae, except for the crickets, and we used adults in that case. Adults were interesting from the standpoint that they are feeding and larger, and so we just that wasn't really my choice, but it was the choice of the head of the project, and it worked fine. We were able to take one male cricket, do the PacBio sequencing, and we did several runs with that portion of the cricket, and send the remainder to Dovetail for doing the HiRise. And I believe it was just, I think you guys had plenty of material for that.
Okay, great. So here's a question. When pooling insects for sequencing, have you noticed whether pooling 5 to 10 insects is better or worse than pooling 1,000 insects? Again, this is probably the most commonly asked question or challenge of insect genomes and insect projects. And I think Brenda was lucky in a way in that, crickets are pretty big, right? And so you don't really need to be concerned about that as much. But it obviously is an issue for some projects. And I've addressed this. I've answered a question similar to this in the chat window here. But just to reiterate, if you can inbreed your insects, absolutely you should do that, because then we are able to pool multiple animals without introducing a lot of heterozygosity. But if you cannot pool them, we really need to try and do the whole project from one animal, the PacBio and the scaffolding. And as I mentioned earlier, the main limiting step is the PacBio. OmniC can actually work very well with a very small amount of material. But the limiting step is really the PacBio, where they need at least 8 to 10 micrograms of DNA. If you're able to extract 10 micrograms of DNA from your single insect, then we're probably going to be in good shape. If that's not possible, then we need to, talk with the project managers and our scientists to figure out the next strategy, which might be Oxford nanopore. It could be amplification. If in some cases, one of our customers just have no other option but to use a single animal, and so we have to figure out how to do that. It may even have to resort to short read sequencing because Illumina requires a lot less DNA than PacBio. But we'll put our brains to work and figure out the best strategy based on our historical projects that we've done for similar species. I don't know if you have anything to add to that, Brenda, in terms of, pooling and, I mean, go ahead.
The question was asked whether you pull 10 or 1,000, if there's a difference, and I don't know, but we
Yeah.
tried to get the minimum number of insects that were needed in order to get the amount of DNA that we needed.
Yeah.
And in our case, those tiny little beetles that I showed you jumping around in the video, 10 were enough to get, you know, to get what we needed. In that case, though, Mark, I'm thinking that we didn't do the same 10 insects on the scaffolding, so that could be part of the problem that we're having, getting down to chromosome level in that insect. So.
Yeah, so Brenda brings up a good point. You know, ideally you should do your de novo assembly, like the PacBio step, with the same animal that you do the scaffolding on. That's the absolute ideal. But if that's not possible, scaffolding can still work effectively, even if you use a different animal for the scaffolding step. But there might be a slight drop in performance. And all these things are variable depending on the organism, the genome. So it's very hard to have a cut and dry answer as to performance characteristics of a project. The best thing we can do, and this is why I think our experience is so nice, because we can go back and look at similar species we've done and give you guidance on how we can give you tips and guidance on how you should approach your project. Okay, we're almost at the top of the hour here. So I think we'll stop here. And I thank everybody in attendance for your time today. And like I say, stay safe and healthy. And we do have 4 more of these webinars coming up. Feel free to attend any of those that you'd like. We will answer every single question that's been given to us. And if you don't mind, we'll send out a follow-up email to the attendees with, just with the list of questions so that everyone can see all the answers. And if anyone wants to talk to me directly, my email is mark@dovetail-genomics.com. So I think that's everything. Thank you so much, Brenda, for such an amazing talk. And it's just
Thank
really
you.
exciting.
Did you hear most of it before I got cut off? I can't even.
Oh yeah.
I don't even know what
Well, we missed. If you want to go back to, I guess you can't now. You don't have your slide, but you were at the acknowledgements section. So if
okay.
there's anyone else
That was basically it. Yeah.
Okay.
That was it.
Great. Well, this has been really fun. And again, thank you for all the attendees. Thank you, Brenda, again, for an amazing talk. And thanks everybody for participating in the poll. We'll make sure that everyone, all that information is used effectively. Yeah, have a great day, everybody.
Thanks. Bye.
Take care now.